This is, true pleasure to introduce Dr. Matthew Anderson. He's the co-director of the Oxford Harrington Rare Disease Center. Prior to joining us in June of this year, he served as vice president and head of neuroscience at Regeneron. His research accomplishments include developing the first genetic mouse model of a frequent form of autism, discovering immune dysregulation in autism and obesity, and uncovering how human-specific retrotransposons regulate brain development and evolution. I don't think you'll be talking about acetaminophen in your talk, I don't think, but I'm excited to introduce him today. Thank you.
Thanks so much. This is really a pleasure to talk to everybody here, and it's great to follow Molly, in fact, because I think it sets it up really nicely, what I'm going to present to you. And I guess I'd like to start by saying that, if you think back in the 70s, an elderly individual in your family starts to forget things, it was labeled dementia, senile dementia. And actually, it really wasn't until kind of much later, it seems fairly recent, that we realized there's a whole diversity of things that cause dementia. And in fact, those are the things that we now direct our treatments at. And I had the honor of leading a whole pipeline of therapeutics for neurodegenerative diseases, including Alzheimer's. And I do truly believe that they're making a lot of progress there. But what it points out is that there are many diseases of the brain that we actually don't understand what's causing them underneath. And I'll give you an example of this, but suicide itself, we really don't know. And I'm a believer now that if you look, you'll find things. And I'll show you an example, but I'll just tell you another example. We looked at cases of obesity. And the genetics of obesity really has helped us understand the circuitry driving that phenotype. And we saw T-cell infiltrates in about 40% of cases. And it's brand new, we did publish it, but it's led me to think that there might be many unrecognized mechanisms that we could actually target therapeutically. Okay, so autism spectrum disorder, we have some of the most significant progress in our understanding of human behavioral mechanisms, I would say, through our genetic insights in autism spectrum disorder, and I'll talk to you about some of those. I actually got my start in cystic fibrosis, a pediatric genetic disorder. It was around kind of a heyday of human genetics. And it really has, kind of been an inspiration for me in therapeutics development. Because they're now, they've extended the life of these individuals from the teens to their 70s. This is a prime example of the true possibility as we understand the mechanism of a disease. Okay, so autism, kind of amazingly, the incidence is just on the rise. It's all the way up to 1 in 31. Now, it's certainly diverse in the severity, but 25% of them are profound. There are more males than females, which again is a puzzle we haven't really solved. I would say a strength, and some people say a complexity of autism is the diversity of genetic causes that have been defined. But the big plus is that it's been a big, intense effort of many talented geneticists in identifying the genes that are affected. I'll show you in a couple examples how we sort of use these pathway and protein interaction network approaches to solve pathways. But it really only accounts for about 10 to 15%. And I had the pleasure of I'm a neuropathologist by training, so I make diagnoses through a microscope. And I was the only neuropathologist that served on a brain banking program for autism that was nationwide. So I looked at all the cases where they wanted a neuropathology diagnosis. And I'll show you in a minute what we found. Previously unrecognized neuropathology, we think it's about 65% of cases. So what I hope is that this becomes potentially a therapeutic target, a little bit like Alzheimer's disease. Core diagnostic findings by definition, and again, this is a behaviorally defined disorder. It's not, there's no blood marker, there's really not very little that we know. Impaired sociability, social communication, and we've done some stuff, I'll show you a little bit of this on the social circuits that we use the genetics to define. the things that make you social, things that if you shut them off, you stop socializing. Increased repetitive behavior and restrictive interests are a feature that's in the models that we've studied. Kind of surprising that you can actually see all these things in a mouse, but you can. Irritability, sometimes even aggression, which is very difficult thing for families. Surprisingly, the mouse model we've studied shows this phenotype, and we are mapping out those circuits. Again, remember, in the obesity example, the genetic mapping to circuits really helped us know where to look pathologically for other causes. Epilepsy is a variably common coincidence, and sometimes even sudden premature death from it. In the epilepsy and there's a convergence of these pathways into the social circuit. And then finally, intellectual disability is variably present, and surprisingly, some of them have almost savant skills. And you may even know of other families with individuals affected by this because it's so common, but some of them are exceptional in very restricted domains. And this really kind of inspired us a little bit in having to find a genetic form of autism that really led us into brain evolution. And it's a gene uniquely in human, chimp, gorilla, and orangutan, and it involves retrotransposons. I'm not going to have time to go into the details of this, but we think that this is a really exciting new direction. So let's go to the pathology of autism. Is there such a thing as a pathology? Some people think that the neuropathology of schizophrenia is kind of like the graveyard of neuropathologists, in fact. But I think that we just didn't really know how to look at it or what to think. So there are two immune systems, the innate and the adaptive, and I just wanted to remind people about what the adaptive is all about. And this is, anti-cancer immunity, but also autoimmunity. And the T cells will attack the cell, and they release substances, and this is what it looks like. But what's interesting is when you really look at an actual tissue where a cancer cell is attacked, you get these structures called blebs. And essentially what it is that there's calcium influx due to these pores, and annexin kind of forms this thing, and it's a way to keep the cell alive by releasing this destructive part of the tissue. Okay, so just keep that in mind. This is our publication now, 2019. Is there a neuropathology of autism? There had been data showing that there's an activated innate immune signature, both protein and transcriptional levels, 2005, 2011, for me, very influential papers, because I had this in the back of my mind as I was looking at these cases. And if you're not a pathologist, you may not recognize what we're seeing here, but essentially, what you can see are T cells. These T cells here in the controls, you can see very few of them in the wall of the blood vessel, but you can see very dense populations of T cells in a couple autisms that are sporadic and a couple genetic autisms. So we were intrigued, and actually it was important to have looked at a lot of brains because you could easily pass over this and really not notice that it was an abnormality. The other really bizarre thing that we saw was shown here. Kept seeing it intermittently in various cases where these eosinophilic kind of pink blobs that were in the cerebral spinal fluid around the blood vessel, that's where they are there. And you can see some of them coming off the wall of that barrier between the CSF and the brain. It turned out it was coming from the astrocyte, glial fibrinolytic protein was positive, and other markers were positive as well. And it was predominantly CD8 T cells, and you could see those in a proximity to these blebs that are a little bit hard to see based on the staining technique. The amount of lymphocyte increase across cases correlated to the amount of GFAP, so it suggests there might be a causality relationship there. And this led us to this model here, which is a new kind of emerging pathology of autism. There are other T cell attack disorders of other organ systems that I see all the time, polymyositis in muscle, for example. But here we think the T cells are attacking the astrocyte glial limitans barrier, and that membrane evaginates off when they're attacked. Is there evidence in the clinic? Well, in a series, I think 3 different studies, people have looked at GFAP in the blood, and this is something you use in Alzheimer's, and it is elevated and quantitatively correlated to severity of symptoms, in fact. So this is the very beginning of a pathology of autism, a fairly large proportion if it turns out to be true, but it's grounded on an innate immune signal that had been already reported by multiple people, and this is the pathology. So, we hope that this becomes the motivator for biomarkers in the blood that we could ultimately use, and then those would serve as substrates to understand whether immunomodulatory therapeutics are effective, hit the biomarker, maybe even the behavioral symptoms. What about the genetic autism? I think that these are really important things to look at. They serve as ways to understand mechanism in much more detail. In Alzheimer's, there's a lot of genetics and a rare subset, early onset, but those are all key players in the pathology. So we focused on this one subtype. It's a cytogenetic abnormality, Dup15q, where the prevalence of autism, again, behaviorally defined, is the highest in these various things. It's extra copies of a certain chromosomal region. I'll show you in a minute. We honed in on a particular gene, UB3A, and I'll show you why. There's a disorder where there's maternally inherited deletions or mutations of UB3A called Angelman syndrome. And they have a whole range of brain-based problems, as you can see. But one intriguing aspect is they have increased social behavior. And as I said earlier, autism is impaired social behavior. This IDIC15 or Dup15q, again, always inherited from the mother. There's an extra little piece of chromosome. They, in fact, have the reciprocal opposite. They have decreased sociability. And variably aggression, some in subsets might be reciprocal with Angelman's as well. But we really honed in on the social behavior, and we in fact showed in a mouse model where we delete the maternally inherited copy. The mice are hypersocial. They're as hypersocial as if you isolated a mouse. You put a mouse in a cage by themselves, and then they socialize. They're very social, hypersocial. So it's like homeostatic, our social behavior. I think that's true in humans as well. So if you get a lot of social interaction, you have to take a break every now and then, at least in my experience. But interestingly, if you overexpress UB3A, which is what happens here, this was our substrate. We recreated all these phenotypes, impaired social behavior, increased repetitive behavior. Ultimately, we found aggressive behavior. People have found seizures, all the things that you can find in the disease. And we've been using this to map out the circuits. And I'm just going to show you one example that we've already published. And again, kind of taking this approach where, the power of large data sets, the genetics of autism is tremendously rich. And, we knew if we could take UB3A and link it up to these other genes, we might find a pathway that could be a therapeutic target. We mapped, I'm not going to go into the details because I don't have a lot of time, but we essentially mapped UB3A's effects to the nucleus. Therefore, we thought it was a disturbance in gene expression. Then we said, well, which of the 600 genes that were being changed could be the key ones? And these things work in complexes. They're little micromachineries typically. We took the autism genes, we took the UB3A regulated genes, and that's what that network is. And it really honed us in on that transsynaptic complex, Nerexin 1, cerebellin 1, and GRID1. Nerexin and GRID1 are both deleted in autism to cause it, and UB3A is downregulating cerebellin 1, and it breaks a synapse, in fact. And in fact, if you look at the copy number variation paper in Nature here, Nerexin 1 is the second most common cause of autism in this case series after ours, the one we're studying. And you can see GRID1, that binding partner of the target of cerebellin 1, is in there as well. I'm not going to have time to talk about it, but this just so happens to be the gene that we think has provided insight into kind of a human evolutionary mechanism. What, allowed the human brain to achieve the high level of intelligence and motor function and things like that. We're very social, really distinct from our current nearest living relative. And then finally, and this is the very end, sporadic autism, can we link it into some of these genetic things? And it just so happens there's a shared molecular pathway. People have looked at, others, not us, have looked at the transcriptome of the brain. They had the inflammatory signal, but they looked at DUP15Q because there's quite a few cases. And the quantitative change of differentially expressed genes in DUP15Q and autism, either down or up, actually kind of are the same. They're going in the same direction, but the magnitude of DUP15Q, the disorder we're modeling, is elevated. So it's a stronger version of possibly the sporadic autism. This is one gene that shows a really dramatic change, S100A8 and A9. Sporadic autism, twofold, about fourfold in DUP15Q. And sure enough, the mouse model that we created, that we published in the supplementary data, actually it's not in the main text, is S100A8 and A9 are the strongest upregulated genes. So I think that this is a model that might have utility in this sporadic autism that we're starting to understand a bit better. And there's a pathway there that one should consider in this work. So this is kind of the end. I'm not going to get into it, but we have a paper out on the aggression circuit. Just so happens that an existing therapeutic that's effective called risperidone for irritability aggression has effects in a circuit that we've defined in using all this conditional genetics and stereotactic stuff and on-off switches to turn on and off the brain. So I'm not going to get into detail there, but we think that the genetics really gets you to particular circuits for irritability, circuits for sociability. We have a new motivated behavioral circuit, the one that causes opiate addiction, cocaine addiction. It's a reward system, and social interactions are rewarding. We mapped social deficits to a circuit that is this reward circuit, and if we turn it on in a mouse in vivo, the mouse is hypersocial, and it fixes the problem. If you turn it off, the mouse sociability is completely gone, actually. So we think that that's something, a toggle switch that one might be able to augment that phenotype. Intellectual disability motor deficits, where we know where to look for these things, but we haven't had the time or the bandwidth yet to go after them. Epilepsy is another really important thing to think about. We heard a little bit about the rewards, the suicidal behavior. It just so happens that there's a punishment circuit that inhibits the reward circuit. They're reciprocally regulating each other. That's relatively new, that we understand that there is such a thing as a punishment circuit. Where might punishment, a depression, even suicidal thought come from if you imagine an activation of a circuit in the brain aberrantly? Maybe the punishment circuit. That's a hypothesis, and we're going to look at the pathology of that if we can get to that point. And I mentioned obesity. They understand the circuits of that quite well. And sure enough, if you use the genetically mapped circuitry and insights, you can find a pathology that can account for a large proportion of cases. And we have something in the works in obesity. Again, I think it's really important to look at the brain, the organ that we want to fix with these therapeutics. And I just shared with you kind of a new pathology of autism. And then, there's many targets and approaches that this kind of research could inspire. And then what makes us human? Again, there's not enough time to really get into it, but we think it involves the retrotransposons that are, many of which are human specific, that have specialized in regulated gene expression in a patterned way in the brain to specialize our brain to achieve what we can do. So this is largely work I did in Boston because I've only been here about 8 months. But you can see all the hard work and the many people that have participated. Thank you.
That was really great. So time for 1 or 2 questions. One in the back.
This is such cool work. I'm very excited to see what you do next. The CD8 T cell pathology, 65% associated with 65% of idiopathic autism is amazing. It's such a heterogeneous disease. Is there information or a sense if that is different than the immune system interaction or the immune pathology that we think is probably involved in a lot of neuropsychiatric disorders? Is it unique to autism?
It's yes. So the pathology of autism is attacking the glia limitans. That's everywhere in the brain, just like white matter in multiple sclerosis is everywhere in the brain. And you get these small little foci of attack. You know, so that's what we think. And the other psychiatric illnesses, we actually don't fully know. You know, there's hints and things, but, you know, obesity, we just published T cells. We think when T cells go into an area, they cause hyperexcitability of the circuit. So if you put some T cells in around the feeding circuit, that act area is going to be hyperactive. You can get epilepsy this way too. And then you're going to eat excessively. When you're not even eating, you're thinking about eating, you know. So it's a kind of a food noise that people describe. I believe that there's going to be many mental illnesses where this mechanism occurs because we really haven't looked at those parts of the brain. There's very few neuropathologists out there, honestly. And seldom do you get a case like these in their hand, and are they going to look throughout the whole brain? No, they're not going to.
One more question, and I encourage you guys to speak to him after we do this.
So I'm just interested in whether you see the male bias in our mouse models.
This genetic disorder does not have male bias, interestingly. So it's a great question. You know, if you look in a fine, detailed way, you're certainly going to see some differences in the phenotype set. You know, you know, where does male? There are some things that are hints at what causes male bias. And, you know, it's kind of complicated to get into them, but I have some ideas around that in terms of this immune stuff.
So one quick idea, which one would be your top idea for the male bias?
Honestly, I like the idea of autoimmunity during pregnancy, in fact, maternal fetal autoimmunity as a potential mechanism. And we've made a model where we immunized a female mouse before she became pregnant with a cancer antigen, which is a very common immunity that exists in the human population during cancer. We think that can't, you know, everybody's heard about paraneoplastic autoimmune disorder, right? Where it's attacking the cancer, but it also attacks another organ system. This is sort of analogous to this concept, and we've got the founding data that has begun that. But of course, remember, this is all adults with immunity, so this would have to carry all the way into later in life. It's a little bit complicated to think about.
Thank you.
So, guys, we're going to be round of applause.